tongue, bro. It's German for F, bro.
I'd like to see it. Yeah, what's the difference? Why don't we get
It's
some
fire.
impact?
I'm not even mad I got hacked, bro. I got some fire songs on Spotify now.
Okay, today's going to be a really, really short lecture because I'm going to teach you how to do the Hardy-Weinberg principle and give you plenty of class time to practice it. So if you have questions, I'm here and we'll do a few examples together too. But today we're going to look at genes within populations and how scientists study alleles and how alleles can change over time in certain populations. So this is kind of our segue into evolution, our evolution unit, talking about genetic variation. There are differences between individuals of a population, and sometimes these differences can be beneficial. So this genetic variation is basically the raw material that natural selection works upon. The definition of evolution is simply how something changes over time. Darwin, in his book of On the Origins of Species, never used the term descent with modifications that sometimes pops up in biology books. But he basically just said that through times, species do accumulate enough differences that make them different species. And so, in a way, this is how new species arise from existed ones, given if they're isolated or separated from that main group. So here we have variations of giraffes and variations of a type of moth, and they're all considered basically the same species, but there are variations. So you can see that you may say that they're different species, but they're not. So natural selection is the mechanism that drives evolutionary change. When Darwin presented his idea of evolution, people thought this is crazy until they figured out the mechanism of what natural selection really is. So this is what drives evolutionary change. And then, like I said, it wasn't really fully accepted until we stumbled upon Gregor Mendel's work with how things are inherited in a species. So here's the definition of natural selection. Some individuals in a population possess certain inherited characteristics and then produce more surviving offspring than individuals that don't have those characteristics. So it gives them a slight advantage in their environment. It gives them a leg up among their peers, so that way they may, therefore maybe survive, pass on that trait, because it has to be able to be inherited. Yeah. Here we have some pictures down here with Darwin's finches. Again, we'll look at Darwin's finches more in depth, I think second semester, like right away, second semester. In fact, you'll actually analyze data from Darwin's finches. I'll teach you how to use Excel and do all the really cool formulas, and basically you'll be like Mr. Rourke. He's pro at it. So, here we have different marsupials and how they are basically just ancestors and how they've just all derived and branched out from one common ancestor. Here is a picture just showing an individual, and then mutations occur, and mutations can cause different variations, and if it's beneficial, they'll go on. More mutations occur, if not, they'll die, and so on and so forth. And so this is kind of how the flowchart of natural selection with evolution works. Okay, now Darwin was not the first to propose that evolution can occur. Jean-Baptiste Lamarck thought the same, well, not the same thing, but some. Oh, I gotta zoom in. I'm sorry. Sorry. It's a Monday. Okay, like I said, Darwin was not the first to propose the process of evolution. Jean-Baptiste Lamarck thought that species could change, but he thought that they could inherit the difference during their lifetime. So we call this inheritance of acquired traits, where here we have a short-necked giraffe ancestor, and if he just continued to stretch his neck during his lifetime, well then longer necks will appear each generation, basically. So he believed individuals passed on to offspring the bodily and as well as behavioral changes acquired during their lifetimes. This is different from Darwin. Variation is not created by experience, but results from pre-existing genetic differences. So Darwin talks a lot about in his book On the Origin of Species, genetic differences and how it occurs before the embryo happens. So the question becomes, how do we monitor population changes? If you're looking at a group of, I don't know, like like I said, finches, how do you monitor the variation differences with each generation? You have to look at the allele frequency and how they change from one generation to the next. Allele frequencies can change based on mutations, which are random, okay? Migration, movement of the same species, into a population, or even, out of a population, which could be kind of bad. Chance events, like catastrophic events. It also could be, like Darwin, when he was on the Galapagos Islands, he hypothesized, how did these animals get here? Here we have a group of islands literally several hundred kilometers away from the mainland. How did they get there? And so maybe there was a huge storm, and these birds got taken up in the current and traveled heck a lot faster than they're used to, and then all of a sudden found the island and inhabited it. So evolution can result from any processes that cause a change in their genetic composition. And so here we have, they look like gumballs, but they represent different alleles. So we have blue, yellow, green, pink, I think that's red. And you notice that they all have the same allele frequency of 0.2. But then something could happen where one allele decreases and the other ones could increase or stay the same or just change. And so this does happen. Alleles do change within a population. And what changes it? Well, there could be many, many factors. So this brings us to population genetics, looking at genes in a population. So you can kind of think of all the alleles in a population as like a giant gene pool. And basically, you're just looking at those alleles. And so I'm going to teach you and show you how you can calculate which allele is more common than the other. This kind of goes back to 7th grade. Actually, I guess biology 10 too, where you know that alleles, when you have two alleles, let's just say the ability to roll your tongue, you could be big R, little r, or big R, right? This is a genotype. It codes for a certain phenotype. In this case, phenotype, which is what you can physically see or what it codes for, so the ability to roll your tongue or not. So genes do influence different blood groups. We'll take a look at some sample problems of ABO, multiple allele blood groups, and they do influence genes, and you already know that because we spent an entire unit on that. Okay, so changes in allele frequency, how can we monitor that? So Darwin and others did not know how genetic variation worked. They knew that something was being inherited from parent to offspring, but they didn't know what it was. They didn't even know anything about the chromosome. Mendel performed his experiments at the same time, but remember, his work was unnoticed for like decades. A lot of people believed in blended variation, where if you took like a red flower and a white flower and you crossed it, you got pink flowers. In some species, that's the case, but in most species, that's not the case. So a lot, at the present, people just thought that you were a mixture of the parents. But if this were true, the effect of any two variants would quickly disappear in subsequent variations, which means that within species, well, we have a species, they mate, the offspring, look like the parents, maybe with some slight variations. They don't look something completely different. So that's where that discrepancy comes in. So why did genetic variation persist, and why did a population never comprise solely of dominant alleles? Because you know that you have the dominant allele, that's the trait that you will express. Well, the Hardy-Weinberg principle will show you, it will describe that. The original proportions of genotypes in population will remain the same from one generation to the next, as long as these 5 assumptions are met. And these are the 5 assumptions: that there's no mutation taking place, that no genes transferred to or from other sources, so there's no immigration or emigration. Random mating is occurring, there's no sexual selection. Population size has to be large, and then no selection occurs, which means no natural selection. If, for example, you go from, I'm going to go back to tongue rolling. Let's say that for homozygous, tongue rolling is 0.25, and heterozygous tongue rolling is 0.5, and the ability to not roll your tongue is 25. Let's say this is the first generation of people, and then I look at, the next generation, and it stays the same. Then these assumptions are being met. If, however, in the next generation, we'll say this goes up to 0.3, and this goes up to 0.6, and all of a sudden this drops to 0.1, so that's equal 1. Well, now I can say, okay, something's going on here. Either we have an influx of people that can roll their tongue and they mated, or there's no random mating occurring. Maybe people are more selective. Like if you can roll your tongue, that might be a good trait to have. I don't know why. I'm just going to stop it right there. So like one of these assumptions is not being met, and you know that something's happening where the alleles are changing in a population. So a little bit about Hardy, Weinberg. Instead of thinking about the consequences of mating between two parents with a specific pair of genotypes, remember our Punnett squares, they wanted to know what happened to an entire population. So like with Punnett squares, you're looking at mom, dad, possibility of offspring. But Hardy and Weinberg wanted to look at an entire population instead of making like thousands of Punnett squares. Is there a way we can look at an entire population of alleles? So they imagined that all the gametes produced in each generation go into a single bin that they called the gene pool. And if I go back a couple slides, you can see the warthogs and their genes going into the gene pool. Imaginary, okay? To determine which genotype would be present in the next generation and in what frequency, they had to calculate what happened when two gametes were plucked at random out of the gene pool. And so the simplest situation possible is that two alleles code for a gene. We're going to be using the letter P and Q in the equation. Your P plus Q equals 1. So P represents the dominant allele. In this case, maybe I'm looking at B or a capital R. The lowercase letter for the recessive allele is Q. So number of dominant alleles plus number of recessive alleles should equal 1 in a population. Okay, so now I'm going to get into a little bit of math.
How is that possible? So it means we do half of an allele mean or like 100%?
It's gonna be percents. It's gonna be decimals, like 0.5 plus 0.5. Yeah. Now the Hardy-Weinberg equation with two alleles is a binomial expansion. So if I was looking at 100 cats and 84 were black and 16 were white, well, I know the phenotypic frequency is 0.84 are black and 0.16 are white. Everyone with me so far? I just turned them into a percent. We can deduce the underlying frequencies of the genotypes for big B, big B, big B, and little b, just with this information. So I'll show you how. We're going to assume white is homozygous for little b, and if you are black, you are either big B, or big B, little b. So with our, going back to our Hardy-Weinberg principle, P plus Q, dominant alleles plus recessive alleles equals 1. Now this only works for the two alleles. But we actually have 3 different genotypes, right? Little b, big B, big B, big B, little b. So how does that work? All 3 of these genotypic or genotypes have to equal 1. So now we have to go into a new little like equation here. So here's my Punnett square. And back to the cats, I know that 16% were white. Now in my Punnett square, where do I find my white cats? On the bottom right corner. Oh my gosh, I can't talk today. Okay, so I'm just going to put a little 0.16 there. I know for a fact that 0.16. Now the rest of the 3 squares is 0.84. I'm going to give you a new equation. So this is the binomial, whatever I just said. Right? P squared, that's my homozygous dominant, plus 2PQ, that's my big B, little b, because I have 2 of them, so I'm going to multiply it by 2. And then Q squared is my little b. So 0.16, what's the square root of 0.16?
0.04.
Just kidding. It's 0.4. Okay, so I know that my little b is 0.4. Can I figure out big B?
0.6.
0.6 because P plus Q equals 1. So 1 minus 0.4 is 0.6. Now can I fill in the rest of my Punnett square? Yeah. Right? So 0.6 times 0.6.
0.12.
0.36.
0.36. Not addition, multiplication.
It's been a rough
0.6 times 0.4.
0.24. I got her down now.
And this will also be 0.24. And it should equal 1. I'm just going to make sure it does. 0.36.
Oh yeah, that's
Oh yeah, it does. Perfect. Cool. I think it's cool. Okay, so I got some more here. 0.6 square root of that is 0.4. Because p plus q equals 1, then p would have to be 0.6, which is the frequency of the big B allele. So here's another example. I'm just going to write Hardy Weinberg up here.
I would just be like, I don't understand this. Which I already don't, so just imagine.
In our imaginary wildflower population, red flowers, big R, are dominant over white flowers, little r. In a particular population, the big R genotype is 0.64. Calculate the P and Q values as well as the genotypic frequencies. So, to tackle this, I would start by making a Punnett square. Right? Okay, so we know that big R is 0.64. So can I figure out my R frequency?
0.8.
It'd be 0.8. So you take the square root of 0.64 and you get 0.8.
And then little r has to be 0.2.
Yep. So then for my frequencies, I go big R is 0.8, little r is 0.2. But then they also want the genotypic frequencies. So for big R, they gave that to me, 0.64. And I got to figure out the other two.
Isn't it 0.16?
0.16. Oh yeah, but then you take it times 2. Yeah. So big R, little r is 2pq. So 2pq is 0.32. And then 0.2 times 0.2 is 0.04. Any questions about this? Pretty easy. So you basically have to find homozygous of 1. I usually always try to find the homozygous recessive first.
Because 8 and the 2 have to equal 1.
Okay, here's another one. So a rather large population of squirrels have 250 red-sided individuals and 750 tan-sided individuals. Assume that red is totally recessive. Please calculate the following. Allele frequencies and the expected genotype frequencies. So I have 250 red-sided individuals. Can I figure out the percent?
0.25.
Yeah, because the total is 1,000. 0.25 for little r, little r. Okay, so I make my little Punnett square. Okay, so then what's my allele frequency of little r?
0.5.
0.5. So if I fill this in, it's all 0.25. Okay, so the allele frequencies for each allele, big R, 0.5, little r, 0.5. The expected genotype frequencies, big R, 0.25. Big R, little r is 0.5. Little r, 0.25. Yep, 1 to 2 to 1. Yep. Conditions happen to be really good this year for breeding and next year. There are 1,400 young squirrels. Assuming that all of the Hardy-Weinberg conditions are met, how many of these would you expect to be red-sided and how many tan-sided?
Well, they're both 50.
Right, so I would start the homozygous. Okay, so 0.25. So you just go 0.25, right, times 1,400. Everyone with me so far? Because they're saying that Hardy-Weinberg conditions are met, so I can still use the allele frequencies or the genotypic frequencies. So it's 350. Sure. So 350 will be red, and then you could just take 1,400 minus 350 to get your tan.
Oh, so that's all you have to do. It's just the same. I thought
Yeah.
it was like you had to figure out like the next gene pool's ratio or something. And I was like,
No.
how's that gonna...
They might. For some problems they might. So okay. So we can use Hardy-Weinberg today, and in fact, scientists do to study the gene pools in certain populations. It's evidence for evolutionary processes. If the assumptions hold true, the allele and genotypic frequency will not change from one generation to the next. And then this is just and if it does change, then you look at one of those 5 assumptions and you try to figure out which 5 is occurring. So, all right, one more example. So I'm looking at a genotypic frequencies of big B. They should be 0.25. For the homozygous, they should be 0.5, and little b, 0.25. If you're looking at a population and all of a sudden you see these genotypic frequencies, where it's like they're favoring the homozygotes instead of heterozygotes, your next question would be, what's causing the excess of homozygotes? Is natural selection favoring homozygous over heterozygous? Is there an influx of homozygous from outside the population? Are individuals choosing, is there sexual selection occurring? So, yeah. Okay, so I'm going to end this.